Hello, fellow microbiome classmates and members of Peer Pod 9. My name is Kate Wright, formerly Whetstone, and today I will be presenting Follow Your Gut, Unraveling How Social Networks Influence Gut Microbiome Composition. The paper that this presentation is centered around can be found within the 637th volume of Nature's Journal and is entitled Gut Microbiome Strain Sharing Within Isolated Village Networks. It was published on November 20th of last year by authors Francesco Beghini and colleagues. Below the citation I have in the upper right-hand corner, I have listed topics that I will be highlighting as we go along. These include bottom line up front, some background information on this topic, the methods used to conduct this study, what exactly did the researchers find, and where do we go from here. And with that, let's begin. Starting off our presentation for today, let's discuss the bottom line up front. So, what's the question being asked with this paper, and ultimately, why do we care? There are so many social interactions one can encounter in just a single day. Waking up in the morning and kissing your husband before heading to work, shaking hands with a colleague to congratulate them on a job well done, sharing a lovely dinner with a group of friends to have the perfect end to a perfect day. While the size of one's social network can vary based on a variety of factors, at the end of the day, we all have one. The authors of this paper were interested in understanding the relationship between one's social network and the resulting gut microbiome compositions. How similar or different would those compositions compare to those compositions of family, of friends, of acquaintances, of people in entirely different communities? What Beghini and colleagues found was that both species and strain level sharing can be seen among different types of relationships that go beyond just familial connections. This includes people living in the same household, being biologically related, as well as more broader connections that extend to outside of the household. Additionally, the occurrence of a connection between two people results in a larger association with strain sharing than what we can see when compared to other factors like diet, age, education, or sex. Another finding of this study is that microbial strain sharing can be used as a predictor for social connections. The ability of the model that was created was improved by including socio-demographic covariates. All of these findings will be addressed in further detail as we discuss the results section later on in the presentation. For now, just sit tight and know that we will be coming back to this topic after we discuss some background information and methods for this study. For this portion, I'll be discussing what we already know about this topic and what's the need for this paper. A 2023 article from Vallis Colmore and colleagues investigates the person-to-person transmission of the gut and oral microbiomes. They illustrate the importance of the maternal microbiome and influence on the infant, as well as the influence of cohabitation on increased strain sharing. For the conclusion of their study, Vallis Colmore and colleagues note that there is a need for reevaluation of the study findings and, quote, accounting for transmissibility and social network structure will improve the design of future studies, end quote. Other articles like the 2019 article by Brito and colleagues, which is referenced within the paper I'm discussing today, focus their efforts on exploring household connections rather than broader searching relationships. They conclude their data, quote, reveals strong transmission patterns most notably within households and between spouses, end quote. Brito and colleagues were also able to use strain sharing to predict spousal relationships only. Another jumping off point for the paper I'm discussing was Dill McFarland and colleagues' article on close social relationships correlate with human gut microbiota composition. Again, like other articles, this one focuses on the familial relationships of spousal and sibling to understand the gut microbiome composition comparison. They identified that spousal microbiota were more similar than that of siblings, especially in sustained marriages. The article I selected to analyze is needed for several reasons. There is a lack of data from non-industrialized world settings, while this study focuses on understudied communities. While previous work focused more on household relationships, Beghini's research spans relationships through entire isolated villages, going way beyond familial connections. Additionally, there is a need for more longitudinal data, which is explored in this piece as well. Finally, machine learning is such a great tool, and Beghini and colleagues explored the applications of strain sharing to predict social relationships. Now that we introduced some of the background knowledge, let's move on to the methodology of the study. Participants of this study resided in Copan, Honduras. According to UNESCO's World Heritage Center, Copan, Honduras is an important site for both Mayan civilization and culture. 18 of Copan's isolated villages were included, and the microbiomes of 1,787 participants were screened for this study. The microbiomes of 301 of those participants from 4 of those villages were remeasured after a 2-year period. The authors noted that all participants in the study did give informed consent to participate in the research, and that both the village leaders and local public health agencies supported the data collection. There's really 2 parts to the data collection for this study. 1, the fecal samples used to identify the microbiome compositions for each participant, and 2, the questionnaires unraveling the social networks between them. We'll tackle part 1 first, followed by part 2. Participants self-collected their fecal samples, and these samples were stored in freezers of -80°C, which is -112°F, until they could be genetically sequenced. The samples underwent shotgun metagenomic sequencing using Illumina Nova Sequence 6000. An image of that device is located to the right of the screen. All reads obtained were screened for human contamination against HG19, a widely used reference human genome. After quality filtering, the researchers were left with 1,787 samples. The researchers note that genetically distinctive strain sharing suggests that the strain came from interpersonal transmission as opposed to environmental factors such as diet. Both species and strain level profiling was conducted using metaphilan4 and strainflan4 respectively. Now that we've discussed part 1 of data collection, let's discuss part 2, social network mapping. In order to map the social networks of participants, researchers pose questions to them like, with whom do you spend your free time? They also ask follow-up questions like how often, etc. I won't read them all to you, but there are more questions found in the supplementary information. 8 relationship types were identified within their study. I made a chart to show the identified relationships and their quantities. The researchers identified 4,658 unique social network links after counting for relationship overlap. This is the part in the presentation that everyone has been waiting for, the results. We'll take it figure by figure and discuss each of the findings. Figure 1 is a 5-part analysis of the strain sharing across multiple relationship types. Part A displays color-coded box plots of each relationship type and their corresponding strain sharing percentage. The median is listed at the top of each box plot. The second to last box plot reflects all pairs of people with no relationship in the same village, and the last box plot reflects all pairs of people in different villages. The highest median strain sharing rate can be found with partners at 13.9%. While the lowest strain sharing rate is 2%, which is for pairs of people without relationships in different villages. From this figure, we can see that the presence of a connection is associated with an increase in strain sharing rate. It is not surprising that mothers share a median of 12.1% of the strains, considering we've discussed in previous journals the influence of the maternal microbiome on the fetus. I want to draw attention to the NSs labeled above some of the relationship types. The NSs reflect relationships that were found to be not statistically significant. What does that mean for the context of our study here today? When something is not statistically significant, it means that there is not enough evidence to confidently say that the relationship wasn't due to random chance. Regarding Figure 1a, this is relevant for partner, same building, child, father, free time, non-kin different house, and close friend. What's interesting about the authors including this in their study is that having a not statistically significant result can be deemed a failure in most scientific reporting. A 2019 article in Nature Journal written by John Brock notes that these type of results are difficult to publish and evidence published in scientific journals is biased towards studies that find effects. So I find it really cool that the authors of this study decided to include these relationship types in their findings, even though it might have been harder for them to publish. And one thing that I want to clarify is that just because something was found to be not statistically significant, it doesn't mean there couldn't be a relationship here. It just means that the findings discovered in this study couldn't provide enough evidence to that effect and were thus inconclusive. However, we did discuss in the background that there is evidence to suggest spousal and cohabitation strain sharing. If we go ahead and take away the relationships found to not be statistically significant, we can still see that closer, more intimate or biologically related relationships lead to more microbial strain sharing. Again, with mothers having a 12.1% strain sharing rate, personal and private relationships at a 9.1%, and siblings at 7.8%. This is higher than the 4% strain sharing rate found with individuals with no relationship within the same village, and the 2% strain sharing rate of having no relationship in a different village. To give some perspective, the authors did mention the average distance to the nearest village is about 1.1 kilometers, and the farthest average distance was 24.7 kilometers. This is 0.68 and 15 miles respectively. Moving on, we see more instances of items not being statistically significant. For example, in figures 1b and c with regards to the frequency of contact and shared meal frequency. We can see, however, that those relationships that encounter each other every day have a strain sharing percentage of 11%, and those that share a meal together almost every day had a 12.5% strain sharing rate. So whether it's generally speaking or through a shared meal, we see that the increase in time that a person spends with one another leads to an increase in their strain sharing percentage. In this case, I actually think that having the not statistically significant comparisons against the strong strain sharing rates of everyday contact and almost everyday shared meals almost gives it more oomph. While the strain sharing after sharing a meal once a month may be due to random chance, the strain sharing after eating together almost every day was found to be statistically significant at that 12.5%. So sharing that time together must mean something, right? Now, some of you may be listening to this presentation and thinking, well, Kate, if people are sharing all these meals together, they're likely eating the same types of foods and sharing the same diet. And if the authors are trying to assert that there's a component to strain sharing that extends beyond the external factors and more so onto actual transmission from one person to another, how does this make any sense? To answer this, I'm actually going to skip over discussing part D of figure 1. I don't really find this data to be too interesting, and since the grading types don't appear to have significantly different strain sharing rates, it's not worth explaining at this time. We're pivoting now to figure 1E. The researchers wanted to account for factors like diet, medications, water source that could be covariates. A covariate is just a variable that has the potential to influence the outcome you're interested in, which in the case of this paper is microbial strain sharing. Figure 1E shows how each covariate predicts microbial strain sharing. On the y-axis, we have all of the covariates, which are further split into all social and familial relationships in blue, and non-kin different house relationships in orange. And on the x-axis, we have the resulting mean squared error, MSE. Now usually we want the mean squared error to be as small as possible, but in the case of this particular graph, the researchers are actually taking the mean squared error permutations minus the mean squared error original. The mean squared error permutations is what happens when the covariates are randomly shuffled, while the MSE original is used as is. To simplify a bit, a lower value here means the covariate wasn't really contributing, and a higher value means the covariate was contributing and thus important in our study. Looking at the graph, it becomes increasingly clear that living in the same household and having a relationship were the most important factors in contributing to microbial strain sharing. Other covariates such as sex, age, water source, diet, and education were not valuable predictors of strain sharing. Another thing that I briefly touched on a bit earlier, but want to go into more detail here, is with the choice to have the covariates split into all social and familial relationships and non-kin different house relationships. Adding the split of non-kin different house relationships shows that even in casual or non-familial relationships, social connection still matters. This is probably the biggest takeaway from the whole paper. However, household sharing is still the strongest factor for all relationships. Well, that's everything that I wanted to cover for Figure 1, so let's move on now to Figure 2. Figure 2 is attempting to see if microbial strain level sharing can be used as a predictor for social connections. The short answer is yes, and when you factor in sociodemographic covariates, that prediction is improved. Part A of this figure is for all social or family relationships, while Part D is only for the village of Ernani. For parts B, C, E, and F, specificity refers to the false positive rate, and sensitivity refers to the true positive rate. The gray line in each graph reflects what the authors call a test no better than chance, which is an AUC of 0.5. AUC refers to the area under the ROC curve. It's just a way of determining a model's overall performance capabilities. The higher the AUC, the better the model's ability to classify correctly, with 1 being a perfect score. There were 4 AUCs accounted for in each graph of B, C, E, and F. Strain sharing rate, only sociodemographic covariates, strain sharing rate plus age and sex, and finally, strain sharing rate plus all sociodemographic covariates. These are indicated on the graph by red, yellow, green, and blue, respectively. For the purposes of this presentation, I will only be focusing on comparing the data between B and E and not C and F. For all social and familial relationships of Figure 2b, strain sharing rate had an AUC of 0.71 plus or minus 0.006, only sociodemographic covariates 0.78 plus or minus 0.005, strain sharing rate plus age plus sex having 0.81 plus or minus 0.005, and strain sharing rate plus all sociodemographic covariates with 0.83 plus or minus 0.005. We can see from the data in this graph that the models were able to use microbial strain sharing data to help predict social relationships. An AUC of 0.71 is an acceptable rating. Remember that 0.5 is like a random guess, while 1 is a perfect score. 0.71 is not perfect, but it's not that bad either. We could consider this to be a moderately strong performance by the model. This is an important piece to the paper. With what we discussed in Figure 1, we saw that a social connection resulted in a higher strain sharing percentage. If there really is a strong connection between one's social network and their microbiome compositions, then we would also expect to see that the similarities in their microbiome composition would also be able to predict the occurrence of a relationship. And this is actually what we're seeing when we look at the graph. We do see a slightly reduced capacity of the model to predict relationships in the non-kin different house graph, with strain sharing rate having an AUC of 0.67 plus or minus 0.007. The prediction ability of the model is increased when we add in the sociodemographic covariates. This is true in both the all social and familial relationships and the non-kin different house relationships graph. This isn't really surprising. The authors even note that it's expected because we do see an increase in strain sharing within family members and cohabitants. So seeing an increase in effectiveness of models including this data makes sense. Like we discussed earlier, the authors were really concerned with ensuring they understood where the strongest predictors were coming from. As with Figure 1e, the researchers used permutation features to understand the importance of each covariate, as displayed in extended figure number 5. For extended data figure 5, the x-axis reflects the different covariates while the y-axis displays the average percentage drop in AUC under permutation. We can see here that after generating 100 permutations, that in both the all social and familial relationships and the non-kin different house relationships, that the strain sharing rate was responsible for the largest average percentage drop in AUC after permutation. In the all social, we see strain sharing rate being followed by household sharing and average age. In the non-kin, we see strain sharing rate followed by average age and sex. What I find interesting about that is strain sharing rate is almost double that of average age when it comes to average percentage drop in AUC under permutation. It makes me wonder if we took out the household sharing in Part A, if the strain sharing rate would appear relatively the same as we see it in Part B. I'd definitely be interested to hear your guys' thoughts on this. The three least influential covariates for the all social were average diet, average Bristol scale, and medication usage. The three least influential for the non-kin were average wealth, diet difference, and average diet. I'm curious why the household sharing isn't the lowest for the non-kin different house relationships one, because the criteria in the relationship is different house, but I digress. In any case, strain sharing rate is the strongest predictor of a relationship when compared to sociodemographic and other covariates. The final figure that I'll be discussing for my presentation is figure 3. This figure is only using 301 samples, which is just a subset of the original sample size. These 301 people were followed up with after a 2-year period to compare the strain sharing between pairs of people across 2 different time points. Ties between people where the average strain sharing rate is above the village median is in blue, where a strain sharing rate below the village median is in gray. While we can see in a few connections the strain sharing rate decreased to below the village median, we also see ties where the strain sharing rate grew to higher than the village median at the second time point. This was really evident in the villages of Mungia and Basuri, and less so in the villages of Arandio and Aspizia. In this slide here, you can see that I've highlighted some key turnover points in time point 2 with these black circles for the villages of Mungia and Basuri. Overall, the strain sharing rate for the majority of ties remained consistent. This suggests that strain sharing is longer lasting and is pretty stable, at least for the samples evaluated in this part of the paper. I do wish that the researchers were able to keep all 1,787 original participants for the section, but for whatever reason they were only able to reevaluate 301. I would argue that there is a need for reevaluation with the 301 at further time points, and possibly reevaluation for participants who were not included in this second round. Increasing the sample size and repetition of trials over time would provide further evidence that strain sharing is stable between connected parties. The last part of figure 3 that I'd like to discuss is part B. Part B shows which factors best predict if 2 people will strain share 2 years later. On the x-axis, we have the standardized beta coefficient. The beta coefficient for this study is a measure of how strongly each factor influences future strain sharing. On the y-axis, we have 3 factors: strain sharing rate at time point 1, occurrence of a previous relationship, and finally the socio-demographic dissimilarity of 2 people at the start, which here is measured by the Malhanobis distance. For strain sharing rate at time point 1, we see a beta coefficient of 0.10. For occurrence of relationship, 0.25, and for the socio-demographic dissimilarity, negative 0.03. This shows that people with social ties have a higher strain sharing rate than unconnected people. The researchers did adjust for socio-demographic similarity in their calculations for that. It also shows that people who shared strains at the start would likely continue to strain share, as evident by the positive beta coefficient. Socio-demographic dissimilarity had a small negative effect. Out of all 3 factors, the social connection was the strongest predictor of future strain sharing. One thing to note is that the wider standard error bar for the presence of a relationship factor. Having a wider standard error bar means that there is less confidence that the estimated data point. This can possibly be explained by the varying relationships one can have. Some are stronger, some are weaker, some are familial, which we've seen increase strain sharing rate, while other casual relationships have less strain sharing. There's potentially more variability for that factor. Even so, if you look at where the lowest point is on the relationship line, it is still above both the strain sharing rate at time point 1 and way above the Mahalanobis distance. So we can see that even at the worst case scenario, having a relationship is still the strongest predictor of future strain sharing. The final figure I will be discussing today is figure 4, strain sharing and social network position. Part A of figure 4 shows the relationship between geodesic distance and median strain sharing rate. Geodesic distance in this study is the number of social steps between two people, meaning their closeness of connection, i.e. first degree equals friend, second degree equals friend of friend, and so on. For this graph, the authors plotted the shortest geodesic distance between two people on the x-axis. On the y-axis, they plotted the median strain sharing rate. The blue line represents the true strain sharing network, while the orange line represents a 10,000 random sample from a within-village microbiome permutation. First-degree connections have a median strain sharing rate of 7.5%, second-degree connections at 5.10%, and third-degree at 4.35%. We can see that people have more similar microbiomes to their first-degree connections than to their second-degree connections, third-degree, or fourth-degree. For third-degree connections, the rate is about at the null network rate, and fourth-degree connections drop to below that of the null network. The null network line represents what would happen if strain sharing was caused by chance rather than social connections. So third and fourth degree connections are actually performing at and then below random chance. What we're seeing is that when people are further out in degree of social network, their strain sharing rate is worse than chance, while being more closely connected leads to much higher than chance strain sharing rates. Part B of figure 4 is a graph analyzing degree of social centrality, which is how many people is the individual in question connected to, against the average strain sharing rate with all village members. The blue line represents the line of best fit, and the gray shading around it is the 95% confidence interval. We can see that people who are more deeply socially embedded in the village, the higher the average strain sharing rate with all village members. The authors note that these socially central people are more representative of the social microbiome, that is, the microbial metacommunity of transmittables strains within the village. Part D of figure 4 is representative of degree centrality against average strain sharing rate with their first degree connections, so not all of the villagers, but only their first degree connections. For this graph, we see a negative relationship between these variables. That means that as someone becomes more socially connected, their strain sharing rate with their first degree connection decreases. If we say that socially central people represent the overall village, it makes sense that they would be further away from their first degree connections. If the individual has a lot of connections, they would likely be the sum of all connections rather than only similar to one person. The authors refer to this as a paradox of popularity. Panels C and E reflect the real social network of the village of Ondarribia. Node color, which is red, orange, or yellow, is reflective of the person's strain sharing percentages. In C, red is for greater than or equal to 4.6%, orange is greater than or equal to 3.9%, and yellow is greater than or equal to 0%. In E, red is 9.7% and orange is 7.3%. The reason for the difference in number is that the values are split into tertiles. Graph C is with all village members, while E is strain sharing rate with only the first degree connections. The size of these nodes reflect the degree centrality. Bigger nodes equal more social connections. In figure 4C, we see that in general, more socially connected people, as evidenced by bigger nodes, their strain sharing rate is more reflective of the village as a whole, and this is evidenced by the red and orange nodes. There is one outlier to this with the large yellow node in the middle, but in general, we do see individuals who have a higher degree of centrality also having a high average strain sharing rate with the village overall. We see a different story in part E. Here, the larger nodes are mostly yellow and orange. This indicates that people who have more connections strain share less with their first-degree connections. What's interesting is that the same large yellow node we see in part C, we still see the same look in part E. That person has a high degree of centrality and little strain sharing with all the village members and their first-degree connections. Their microbiome must be really unique. The paper does go through more figures and analyses, but I feel that the figures 1 through 3 showcase the most important aspects of the paper's goals. The researchers of this paper wanted to better understand the relationship between gut microbiome compositions and the social network as it relates to the isolated village communities in the highlands of Honduras. By presenting data on a subset of the world that isn't your stereotypical industrialized nation, we gain insight into how these communities thrive. We also can see patterns in these isolated villages that might be harder to uncover otherwise. Through their research, Beghini and colleagues found that microbial strain sharing and presence of a social connection illustrate a bidirectional relationship where strain sharing can be used to predict social connections, and the presence of a social connection reflects an increase in strain sharing rate. However, it is difficult to distinguish between a shared environment or transmission from person to person based on observational data collected in this study. On this slide, you'll see some questions that I have for you based off of this presentation. The first question for you is, in this study, human contamination was screened for HG19. Why do you think the researchers chose HG19 instead of HG38? How much impact does this have on uncovering potential contamination? Question number 2. In Figure 1A through C, there are some relationship types that were found to not be statistically significant. What does this mean for the results of the study, and how do you interpret these findings? Is there anything that can be done in the future studies to potentially mitigate receiving those type of results? Question 3. In Figure 2, the researchers used models to see if microbial strain sharing could predict social connections. Why do you think that socio-demographic covariates increased the model's ability to predict social connections? The researchers of the study made sure to collaborate with local health officials and village leaders to conduct this experiment. Do you think it was necessary to involve them? What benefit could including these individuals provide? What does this study make you think about your own gut microbial compositions as compared to other people in your social network? How can you apply the findings from this study to your own life? This final slide here is the references slide. So what I've done here is for the first reference, that's going to be the one for this paper that I've been discussing. And 2 through 6 were used in the background or other pieces to my presentation. So here are all these references. Feel free to look them up and to do your own investigation.